Hello friends, this video on Biotechnology Principles Part 21 is brought to you by examfear.com. No more fear from exam. So with this we have reached towards the end of this lesson. So let us quickly have a look at some of the questions. Question number 1. Can you tell whether enzymes are bigger or DNA is bigger in molecular size? How did you know? So which is bigger, enzymes or DNA? Now what are enzymes? Enzymes are nothing but proteins. And what is DNA? DNA is the nucleic acid. So DNA is the double-stranded structure, and we know as far as size is concerned, DNA is quite long in size. It is so long that it cannot directly fit into the nucleus. So in order to fit inside the nucleus, it is present in the form of a coiled structure. So that means we can say that enzymes are smaller in size than DNA because DNA is very long. So its molecular size increases. So enzymes are proteins which are synthesized from genes located on DNA. So on this DNA only, a certain set of genes, they will code for a particular protein. Again, a certain set of genes will code for a particular protein. And the proteins are nothing but enzymes. All enzymes are proteins. So basically, if you see, proteins are being formed just from one part of DNA. So with that analogy as well, you can assume that enzymes are smaller than DNA. Question number 2. Do eukaryotic cells have restriction endonucleases? Justify your answer. So eukaryotic cells do not have restriction endonucleases. These enzymes are present only in the prokaryotic cells. Why? That's because eukaryotic cells are highly methylated by enzyme methylase. Now what do we mean by methylated? What is methylation? So methylation is a form of alkylation with a methyl group. So what happens in the process of methylation? So let us first try to understand what is methylation. So methylation is derived from the word methyl. What is alkylation? That is adding an alkyl group. So methylation is nothing but adding a methyl group. So methyl group would be CH3 group. So when a CH3 group is added to the cytosine or adenine DNA nucleotides, that is called methylation. So this methyl group will be added either to the adenine base or to the cytosine base. So then we say that the DNA is methylated. Now in eukaryotic cells, what happens is that due to the presence of this enzyme methylase, the DNA is highly methylated, and due to the highly methylated DNA, they do not have the restriction endonucleases. Whereas in case of prokaryotic cells, they are not methylated, and that is why they have restriction endonucleases. So it is all due to the presence of this alkyl group that the restriction endonucleases are present or not present. Question number 3. Besides better aeration and mixing properties, what other advantages do stirred tank bioreactors have over shake flasks? Now, some of the advantages of the stirred tank bioreactors, which we have discussed just now, are as follows. They have pH control system. Actually, these stirred tank reactors, they have a lot of control systems. So you can control a lot of things. So they have pH control system. So that means you can control the pH inside the reactor. You can make the environment more acidic. You can make the environment more basic as required. Temperature control system. So you can increase or decrease the temperature. Foam regulating system. So when you have the culture broth, there will be foam. If you want to increase or decrease the foam, that can also be controlled. Sampling ports to withdraw small volumes of the culture for testing purpose. So here you can see there are ports, or there are opening through which small volume of the culture can be taken out for testing purpose. Now all these facilities are not available in case of shake flasks, and that is why these are advantages. Now due to the presence of so many control systems, you get a lot of control over the bioreactor. So you can regulate it or you can control it as per your wish. So these are some of the advantages which are present in stirred tank bioreactors that are missing in the shake flasks. Question number 4. Can you recall meiosis and indicate at what stage a recombinant DNA is made? Now, whenever we talk about recombinant DNA, so recombinant DNA is formed by recombination. That means combination of two different things. Now, when you talk about recombination, you remember which stage of meiosis? First of all, what is meiosis? It is reduction division, where the chromosome number reduces to half. So, if you start with a cell having N, so having 2N, so you end up getting a cell having N. So your chromosome number reduces to half, and that is why it is said to be reduction division. So in this, crossing over takes place in meiosis 1 in the Pachytene phase. If you remember, the entire process of meiosis is divided into two stages, meiosis 1 and meiosis 2. Again, meiosis 1 has various steps like Pachytene, leptotin, and similarly meiosis 2 will also have the various phases. Meiosis 1 will have various phases like metaphase, anaphase, telophase. Similarly, meiosis 2 will also have. Now, the prophase 1 of meiosis 1 will have multiple stages like Pachytene, leptotin. Crossing over takes place in prophase 1 of meiosis 1. So, meiosis 1, inside meiosis 1, you will have prophase 1, and inside prophase 1, you will have the Pachytene phase. And in this phase, crossing over takes place. What happens in crossing over? The process where genetic material is exchanged between the non-sister chromatids of homologous chromosomes. Now, this is how homologous chromosome looks like. This chromosome and this chromosome. So, these two are homologous chromosome, because both of them will have similar genes on their corresponding locus. So, these are homologous chromosomes. Now, these two homologous chromosomes will undergo exchange of genetic material. So, if you see, these two are going to be the sister chromatids. The two pinks are going to be sister chromatids of each other. Similarly, the two blues are going to be the sister chromatids of each other. But during crossing over, what happens is that the exchange of genetic material takes place between the non-sister chromatids. So, between the pink and the blue, the exchange will take place. Now, once crossing over takes place, what happens? You get recombinant. So these are called recombinants, and this process is called recombination. So if you look at this chromosome, it has pink, it has blue. If you look at this, it has blue, it has pink. So it has got mixture of the traits of both the non-sister chromatids. So these are said to be recombinants. So therefore, recombinants are formed during the Pachytene stage of prophase 1 of meiosis 1. So this is where recombination occurs. Question number 5. Distinguish between plasmid DNA and chromosomal DNA. So when you talk about a plasmid DNA, it is a circular DNA which replicates independently. That is the most important property of plasmid DNA. Whereas chromosomal DNA, obviously this DNA is present as a part of chromosome, so it will replicate as a part of chromosome. So plasmid DNA is extra chromosomal DNA, that is, it is present outside the chromosome, but chromosomal DNA is present inside the chromosome. Now that is why this plasmid DNA acts as a good vector, because it can replicate independently, and that is one feature which we are looking at in a vector. Exonuclease and endonuclease. Now, as I have mentioned before, exo always means outside or exterior, and endo means inside or interior. So these are those enzymes. Nucleases are those enzymes which help to cut nucleotides from a nucleic acid. So exonuclease will remove nucleotide from ends of DNA molecule, that is from the outer part. So if you have your DNA molecule, you will have several nucleotides. Let us suppose each of these circle represents a nucleotide, and one nucleotide will have one sugar, one base and one phosphate group. So this is one nucleotide. Again, this is one nucleotide. Again, this is one nucleotide. So if you see, these nucleotides are just joined to each other to form a polynucleotide. Now if an enzyme starts removing nucleotides from the end, that is called exonuclease. But if there is an enzyme which tends to remove a nucleotide from between, let us suppose it removed this nucleotide, so then that is going to be an endonuclease, because it removes nucleotides from within the DNA molecule. So this is going to cut the DNA at specific sites, and this one is going to cut the DNA at ends. So that is the difference between exonuclease and endonuclease. So with this, we have reached towards the end of this lesson, and I hope that this lesson on biotechnology principles would have helped you to understand the basics of biotechnology. So see you all in the next lesson. Thank you. Please visit www.examfear.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.